Hi everyone, and welcome to today's Daily Booster for Biology. We are now at number 11 in our series, and you will notice the lovely purple box that tells us that this is an advanced information listed topic. Now this is listed on both foundation and higher biology, so it's important for everyone. What we're going to be looking at today then are some of the transport tissues within plants. The first thing we need to know about then is the tissue xylem. Now we need to know what xylem transports, and xylem transports water and dissolved mineral ions. And the easiest way that you can have a little bit of a hint to remember this is think about the alphabet. Water in xylem, W, water in xylem, XY, so WXY, water in xylem. In terms of the direction of movement, the xylem is going to transport water from the roots up the stem to the leaves. So it's got this one-way movement from the roots moving up the plant. Water is going to enter our plant through the roots by the process of osmosis, which we looked at in our previous booster, and the mineral ions will be taken up by the process of active transport. Again, if you're not 100% certain on those, do go back and revisit them. But remember, osmosis, the movement of water from that area of high water concentration to the area of low water concentration across the partially permeable membrane, whereas active transport is going from an area of low concentration to an area of high concentration and does require ATP to do that. So what's special about these xylem cells then? Well, first of all, they are dead cells. Because they are dead cells, then what we have got are basically like giant straws. Now, the reason that they have to be dead is because in order to make it so that these giant straw-like structures can withstand the forces of bending, then we need to strengthen the walls. And we do that by adding a substance called lignin to them. Lignin is great because it provides support, it thickens the cell walls, but it does kill the cells. The other key feature about our xylem vessels is that they have no end walls between the cells. So what we actually have done is broken down any of those individual cells to create one long tube. So dead cells with no end walls with lignin to thicken them. Tissue type number 2 in our plants is phloem. Now the phloem is all to do with transporting sugars that are made in photosynthesis. So throwback to our B1 work on photosynthesis, balanced symbol equation there just to remind you. Now what we actually find is that the process through which we move these sugars is called translocation. So that is the movement of dissolved sugars, and these may go up or down the plant. So this is like two-way movement. Now key areas that we need sugar in our plant, the meristems, which we established are those undifferentiated cells in our plant, which is where we're going to need those sugars to make new cells. Or it could even go all the way down to the roots where we can store it. Think particularly about those plants that use things like bulbs and the like. Great for storage down there. Key things about our phloem cells then. They are living cells. They do still have end walls, but they've become sieve plates. So what we've actually done is punched little holes basically through it to make it so that larger molecules can cross easily. What we then find in terms of how this is all organised, the xylem and the phloem go together in a structure called a vascular bundle. Now the location of the vascular bundle in the plant is all to do with providing support, and different regions of the plant find those vascular bundles in different places. If we look at the leaf first of all, then it occurs as a network and the soft tissue is then basically attached to it to support it. In the stem, those vascular bundles are around the outside in a little ring, as you can see in the middle diagram. And in the root, we find it in the centre of the root, and that allows the root to bend. The process we now need to look at is the process of transpiration. And quite simply, transpiration is the loss of water vapour from the aerial parts of a plant. You could say that as the loss of water vapour from the leaves, absolutely fine. Please be careful to make sure you do include the word vapour when we're talking about the loss of water. Don't just say loss of water from the leaves, because that's not going to be enough. Loss of water vapour from the leaves, or you could say the evaporation of water from the leaves, but you can't just say loss of water on its own. Transpiration stream is actually the movement of water from the roots through the xylem and then out of the leaves. So that's looking at the entire flow of the water, if you like. And what we then do is we set up a system through which we need to constantly replace that water that is lost from the leaves by taking in more water through the roots. So let's take this one step at a time, starting in the roots then. Water enters our plant through those root hair cells by osmosis. And root hair cells are specialised cells that have got that bit sticking off down here, as we can see, and the whole purpose of that is to increase the surface area. If we've got a larger surface area, we can have more rapid exchanges. Once the water has entered the root hair cell, it's then going to travel cell to cell until it reaches the xylem in the centre of the root. Once it gets to the xylem, it then travels through the xylem vessel throughout the plant. When it gets to the leaves, we need to know two key structures. The stomata, first of all, their little pores, and the guard cells, the cells that surround the pores. So what we actually have, the stomata, these little holes, and you can see them on the little diagram on the left there. These are little tiny pores on the surface of the leaf that allows carbon dioxide to diffuse into the plant and water vapour to diffuse out. They are surrounded by these special cells called guard cells, and they're designed in such a way to open and close the stomata based on how much water is present. So do note this inner wall here. So what we can see is on the inside wall of our guard cells, the ones closest to the stomata, they are much thicker than the outer wall. The reason for that is that when there is plenty of water, water moves into our guard cells, they become swollen or turgid, and it pushes out the outer wall. Now, because the inner wall is thicker, it's not going to push that one in. So that means our stomata actually open. So we get that bending effect that means that we have an open stomata. When we don't have much water present, what happens is the cells become flaccid and then they close the stomata. When we consider what happens when the guard cells have opened the stomata, then at that point we have a pore to the outside world. That means water can evaporate from inside the cells to the air spaces. We then set up this concentration gradient because we've got a high concentration of water vapour inside the cell, and then we've got a low concentration outside the leaves. So what we then have is a concentration gradient between inside the leaves and outside the leaves, and as a result of that, water vapour moves down the concentration gradient and will diffuse out of the plant and into the surrounding air. When we lose that water from the leaves, we reduce the pressure in the xylem vessels. So the top of the xylem vessels then has a lower pressure. As a result of that, we've got a high pressure in the roots, a low pressure at the top, and therefore water moves up through the xylem from the high pressure area to the low pressure area. Hence why it always goes from bottom to top in the plant. It's not always the case that we want our plants to lose lots of water vapour. Sometimes we need to retain as much as possible based on where that plant actually grows. So those that grow in very hot climates need to keep as much of their water as possible inside the plant. The way that plants have actually adapted to minimise their water loss is by having this structure called a waxy cuticle. Quite simply, it's a layer of wax that sits on the surface of the leaf, and depending on how thick it is, that's going to determine how much water loss is reduced. So plants in very hot areas where there's limited rainfall are going to have a much thicker waxy cuticle than those where there's plenty of rainfall. The reason that we need to be cautious with how much water our plants lose is because if they lose water at a faster rate than they're able to take it in, then they will wilt. So what that means is they kind of go a bit floppy. Now, when that happens, the leaves are going to collapse, and that reduces the surface area available for evaporation. So it's like a defense mechanism, if you like. As the water levels inside the plant are decreasing, the whole plant then droops and as a result of that we reduce the surface area through which evaporation of the water can take place. So it reduces that water loss. We also see the stomata closing, but there is a downside to this. As much as it's protecting the plant from losing more water, it is going to limit photosynthesis, because when the stomata are closed, we can't take in carbon dioxide, and therefore we're going to run out of one of those key chemicals that we need for photosynthesis to take place. So this is not something the plants can do for a very long period of time. Final thing to do for today then is head on over and have a go at the quiz, see how well you've understood today's topics. As always, if there are topics that you don't score well on or haven't understood too well, then swing on over and have a look at the video on the main channel for that particular topic, go through your revision guides, your classwork, or just ask your teacher to go over it just for your exam. Don't forget to join us again tomorrow for our next Daily Booster.